All right, very good. Okay, so we finished up with the cercozoans last time, and we're going to move on to the next group now that we're going to talk about. And this is the group that actually starts to incorporate some of this polyphyletic nature that we talked about in protozoans. Yeah, there we go. If you take a look again at protozoan diversity, remember, it's an extremely polyphyletic group. As a matter of fact, we don't refer to a kingdom protista anymore. I mean, you will see it written that way, kingdom protista, but in more serious discussions, the kingdom protista is really an artificial designation, a very polyphyletic designation for reasons that we've talked about in class, right? Because if you look at it, in amongst all these protozoans, and the protozoans are all the ones that are in the black type. So the diplomonads that we went through, the algae, the cercozoans, the algae that we'll talk about today, the red and the green, these are all protozoans, but look what's mixed in amongst all of those, right? The land plants, the fungus, and the animals. And I've said this before, among biology, we generally like to look at the protozoans as being the stem group that gave rise to all the other eukaryotes. So extremely polyphyletic, because if you look, for example, among the animals are more closely related to a protozoan known as a choanoflagellate than the choanoflagellates are related to other protozoans, you see. And then among the land plants, we got the green algae and the red algae. They're more closely related to each other than the red algae and some of those green algae that are protozoan-like are related to the other ones. So very mixed up group here among these organisms. We're turning our attention now to this group down here in the yellow, the Archaeoblastida, and that includes the red algae and then the two groups of green algae. Now, this word algae again, let me make sure that we clarify any confusion on that. Something that is an algae can be many different things. If we use the word algae, it could be a prokaryote, blue-green algae. If we say red algae is a protozoan. If we use the word green algae, here's where it becomes even more confusing. Some green algae are considered more protozoan-like. Some green algae are considered more plant-like. Some people consider certain green algae protozoans. Some people consider those same green algae plants. Which one is right? Well, go back to the very beginning of the semester when we talked about what makes a species, right? It's not Aristotle. These things aren't rungs on the ladder. So it's all gradations. But those gradations indicate to us all this commonality of organisms, right? All this relatedness of organisms. And that's why when we take a look at these things like the green algae, in the group of things we call green algae, there's going to be the protozoan-like green algae, and then there's going to be the more plant-like green algae. And I'll get to that when we start talking about those. But I want to talk about one of my favorite group of protozoans first, who do we have come in. Savannah, how are you today? So I want to move my attention now to talk to the red algae. Very cool group of organisms. Red algae are called red algae because, of course, they're red. There's some examples you can see. Nori, the stuff that they wrap around sushi, that's a red algae. That stuff on the outside, it actually has a red color before it's been processed and used for consumption. But these are all examples of red algae. Now, interesting thing about red algae, number one, most of them are multicellular. Yeah, that's something that's unique among protozoans, right? Remember, red algae are considered protozoans. I don't really know of anybody who considers them anything else. So I think these are pretty set into whatever this protozoan kingdom, however that's going to be parsed out. Definitely not a plant, definitely not an animal, but it is multicellular. And again, remember, multicellular doesn't mean tissue. There is a very distinct difference between having lots of cells and having a tissue of cells. And again, that kind of works along a gradient as well. We're going to see examples of organisms that are multicellular, especially when we get into animals and look at like sponges that I mentioned. They're multicellular, but we say in sponges they're actually kind of what we say is a loose collection of cells. So they're multicellular, not tissues though. So more on that when we start moving into the plants or moving into the animals. But I do always want to mention that because I always want you guys to keep in mind about how these little changes occur through time. That multicellularity and having tissues didn't arise at the same time. Things were already multicellular, And then this whole idea of tissues came on the scene. Red algae are oceanic organisms. They're found typically in tropical oceans. They need warm water to survive. Very often, these are the organisms of many times in deep water that you're going to find. All right, which is interesting, because here where we throw a little bit of a catch. Red algae is a photosynthesizer. That's how it survives. It's an autotroph, okay? It's a photoautotroph, so it needs light in order to take carbon dioxide and turn it into food. It's a photosynthesizer. Now, if you look at the chart on the left-hand side, the little chart in the lower left, that is basically a chart that tells you what colors of wavelengths of light penetrate through different depths of water. Like, for example, if you notice the red light end of the spectrum, red light drops out very quickly in water. There's no red light left after 30, 40 meters, something like that. I remember being in the Bahamas one time when I was in graduate school and doing a project, scuba diving. We were scuba diving. And I was down at depth. I don't recall what depth. It was probably 50, 60 feet, something like that. And we were laying ropes to do a transect. A transect is a device used to count animals, basically. And the ropes were going to be sort of our guide that we followed. And we took the rope and we took these spikes and we pounded them into the bottom to kind of lay out where the rope was going to be, and then tied the rope to it. When I cut the rope, though, after we tied it with my dive knife, I nicked my finger. All right, cut my finger. Okay, no big deal. Everybody's done that. What was so interesting, though, when I looked down at my thumb, what was coming out of the little cut in my thumb was blood, obviously, but it wasn't red. It was green. If you're a scuba diver, when you get down, you don't have to get very deep either. Your blood turns green. And the reason why that is, if you remember back from Bio 1, things look the color they do because of the color of wavelength they reflect. Now, my blood looks red because it reflects red light. But down at depth, there is no red light. So it couldn't reflect the red light that never occurred there. It reflected the next wavelength of light, which is in the greens and the yellows, because they can penetrate down a little bit further, too. Now, take a look at the green light. Green light, of course, is the light that's least important to plants. The most important light, now it extends pretty far down. The most important wavelengths to light are going to be up here in the reds and the oranges, right? And, of course, if you remember from Bio 1, when you looked at a spectrophotometer printout of chlorophyll, remember chlorophyll absorbs in two peaks, has two peaks. It was the blues and the purples, but very little absorption in the greens and the yellows. That's why plants look the color they do. They reflect the green light. I always tell my Bio 1 students to get a real sense of how this works. If you put a plant into a room, a dark room, and the only light that was shining in that room was a green light, those plants would die. Because as far as those plants are concerned, they're in the dark. They don't use green light, they reflect it. So now, let me go back to my original topic about the fact that red algae are photosynthetic. Take a look at that light chart down below, keeping in mind the ones I've circled are the wavelengths of light that are most important for photosynthesis, especially the ones up in the red and the oranges, right? That's 680 to 700 nanometers. That's when those photosystems are most sensitive to. How do these things photosynthesize then? Any idea? I mean, some of these can grow at depths well beyond 200 meters. So how do they survive if the only colors of light that are getting down there, 150, 200 meters, are the green light, what they don't need, and the red light, which they really do need, has attenuated or faded out well before you even get to that depth? See, Mother Nature always seems to find a way, right? What these organisms have done living in deep water, being photosynthesizers, they still have chlorophyll. Red algae still have chlorophyll. It's in their cells, but they also have another pigment that's called phycoerythrin. Erythrin means red. Okay, erythrin, like erythrocytes. Gosh, now I'm gonna have to try to spell it. Erythrocytes are red blood vessels. Right? So phycoerythrin is a red pigmented dye. It's what gives red algae their color. When something is red, then that means it's reflecting the red light, and typically what's happened, it's absorbing the other end of the spectrum. So red algae use this phycoerythrin to absorb light, but they don't absorb light in the wavelengths that plants on land do. All right, they can't. Plants on land are using these wavelengths of light. And these organisms, these red algae down at depth, don't have access to those wavelengths of light. So what they do is they absorb what they are getting, the blues, the purples, maybe a little bit of the green, and phycoerythrin absorbs that wavelength of light, absorbs that energy, and then transfers it to chlorophyll. So in photosynthesizers like this, we actually have two photosynthetic pigments working in unison with one another. The phycoerythrin does the absorbing of the blues and the greens, the light colors that get down to that depth. And then, and by absorbing light, of course, it's energy, and then transferring that energy to chlorophyll. And then from there, it works the same way as it does in regular plants. The reason why red algae don't look, why you can't see the chlorophyll is because they have so much of that reddish pigment, that phycoerythrin. As a matter of fact, in the nori, when they process the nori, the fact that you get a little more of a greenish color in there is because a lot of the phycoerythrin pigment is destroyed during the processing, and so what you see in the nori is more of the green color, more of the chlorophyll. Isn't that cool? I love this kind of stuff, man. What an incredible adaptation. If you can't photosynthesize because you don't get the right wavelengths of light, then another pigment could be useful by converting or using that energy and transferring it over to chlorophyll. All right, the green algae. Most of these are freshwater. There's a few marine species. There's even some variations of algae that are found in soil. Oftentimes these are the photosynthetic partner in a lichen. Remember I said a lichen is some photosynthetic cell and then a fungus growing together. That photosynthetic cell could have been a blue-green algae, right? Could have been a prokaryote, or it could be a green algae, right? Green algae is a paraphyletic group. So what we mean by that is the fact that when we talk about green algae, we're referring to green algae as the descendants, okay? But we're not including all of them in our discussion, because remember the red algae belong in this group as well. So the green algae is an offshoot of the red algae group. Now, in amongst the green algae is where the seeds were laid for plants to move out onto land. For example, the caryophytes. Now, Chara, right there that you see in the picture, that's still alive. That's still an organism that's alive today. But caryophytes, ancestors of the one you see in the picture, were ancestors to land plants. About 450 or so million years ago, plants moved from water out on the land for reasons that we'll talk about when we get there. So those were the direct, so caryophytes would be the more plant-like green algae. And coelacanths, these are coelacanths right here, they would be the member of the chlorophyte group. And the chlorophyte group is a green algae group that was closely related to the ancestor of land plants, but not the ancestor of land plants. So think of chlorophytes as being the cousin of the ancestor to land plants, right? So chlorophytes are the cousin to the caryophytes. Chlorophytes are then considered more protozoan-like. Many of them are unicellular. One of the earliest green algae was one similar that we see in the picture there on the right. This is 5 microns, so this is a very small cell. Let's see if I can get this. This is a tongue twister. Chilamadomonas. Chilamadomonas reinhardti. That's what it is. Chilamadomonas reinhardti. Remember, the words in science, the scientific names are very often very phonetic. So if you're trying to figure out how to pronounce them, do it phonetically. Well, it was these unicellular green algae that diverged from the line that eventually gave rise to the caryophytes about 1 billion years ago. So about 1 billion years ago, we have this split up in the protozoan group that occurs, and then the caryophytes went off on their land, on their path that gave rise to the land plants, and the chlorophytes continue along the line of the green algae. Today we have existent representatives of the chlorophytes. The Chilamadomonas reinhardti is one. The coelacanth that I circled in the upper right, and Volvox. Everybody's familiar with Volvox. We were going to look at them under a microscope. Big green hollow sphere. Now, we actually look at Volvox in the plant part because, again, it kind of represents this idea that Volvox are considered kind of more plant-like, even though if you're looking here, they're not in the direct lineage to land plants. It's the caryophytes. Now, these are very interesting because this is a colonial algae. These are, Volvox is a collection of cells from anywhere from 500 to, over 60,000 individual cells that live together cooperatively. Individual cells that live together cooperatively. We're seeing that gradual change, that gradual advancement, if you will, towards tissues. Right? We got individual cells living together. We even had cells living together in prokaryotes when they made biofilms. Remember that? The plaque you get on your teeth? All that increases the efficiency. So, if we talk about the protozoans as being kind of the stem group for all the eukaryotes, the plants and the animals, and especially when the animals, tissues are a very big thing, we see experimentation or organisms that are using this multicellular cooperative approach already. Now, is a Volvox and its cooperative living cells a tissue again? No, it's again a multicellular organism. Now, would it need to evolve and to become tissues to be evolutionary fit? No, you should be able to answer that question very simply. They do quite well living the way they do. So just because we say, all right, these organisms were on the path to multicellularity in tissues, but they stayed down here at this sort of cooperative living stage, doesn't mean that's a bad thing, right? Evolution doesn't have a goal. Evolution's goal is basically you've got to do enough to survive today. And so Volvox, living as these cooperative group of cells, do enough to survive today. All right? Now, some of the things that the chlorophytes, some evolutionary innovations by the chlorophytes, were first of all, what I was talking about, this formation of colonies from individual cells, right? Multicellularity, like for example, ulva there on the left-hand side, sea lettuce. It's very common in Florida. See it all the time. Looks like it's green, very thin. It's almost like just a thin membrane. That's sea lettuce. Sea lettuce is multicellular. Now, what's the difference between being multicellular and being Volvox and being multicellular? It's just a little bit further down the road, right? So you would assume in a multicellular there's even more cooperative behavior, not behavior, but cooperativity amongst those cells, right? Now, among Caulerpa, which is another chlorophyte, we actually get something called multinucleation. In this particular case, what multinucleation, what we're talking about, is cells undergo karyokinesis, but they do not undergo cytokinesis. So we get these supercells. And what I mean by that is we get one cell with lots of nuclei in it. Now, this is going to be something really interesting when we get into the fungus. This is basically how fungus live. Among fungus, the individual fungal cell, that's not exactly what it would be in fungus, but nonetheless, the emphasis is not on the cell. The emphasis is actually on the nucleus. Reproduction in fungi is all about the nucleus. In many cases, there are many fungi who are multinucleate, just like Caulerpa. We call these things supercells because you remember we talked back in evolution early on that there was heterozygote advantage, or there was the advantage of being diploid, right? Holding alleles that might be beneficial in the future. Remember, that's what it means by having two copies of each chromosome. We actually have, perhaps, if you're heterozygous for a trait, a little different between those. And that might be the difference between you being most fit or not so fit in an environment. What we get in a situation with multinucleation that Caulerpa does, and then fungi really do, is we get a situation where the nuclei never fuse. A cell has more than one nucleus in it, each with its own set of genetic information. So it's kind of like doing the same thing as being diploid, right? Holding that recessive allele so that it may be beneficial in the future without actually just fusing the nuclei together. And in some cases in fungi, you'll have two nuclei in a cell. In some cases, it'll be multinuclei that are in a cell. All of the emphasis being on the nucleus where the genetic information is, right? And we'll talk more about those when we get into fungus. Really cool group. All right, now we're going to move out of the Archaeoblastida to the Uniconta. This is the Uniconta, our last group down here. All right, so we see this idea that what we got going on here is these organisms evolving and changing. Some of them are getting closely related to land plants. But now I mentioned that the evolutionary innovations in the chlorophytes were beneficial for land plants. Remember, we had chlorophytes and caryophytes. Being that they're cousins of one another, the evolutionary innovations in the chlorophytes could have been beneficial to those caryophytes that moved out on the land. Okay, because that's where the land plants come from. Now notice first of all in the lower left the dashed line, which means we're not sure exactly how that break occurred. We're not sure exactly how that part of the family tree goes together. So the dashed line represents some sense of uncertainty. The word uniconta tells us something about members in this group. Uni is one, and cont is a projection or a pseudopod or a flagella. Organisms in this group have one flagella or they have pseudopods. Like the amoeba up above is a unicont. They have pseudopods. We belong in this group, animals, because the cells that we make with flagella, you think, what cells do we make as flagella? Sperm, have one flagella, right? And that's the same thing with the fungus too. This group that's the Uniconta really demonstrates the polyphyletic nature of the protozoans because we have the slime molds, the tubulinids, the entamoebas that we'll talk about. We don't talk about the nuclearids. And we'll talk about the choanoflagellates when we get into the animals. But notice these are all mixed in with these groups, the two kingdoms, fungi and the animals, right? Very bizarre group. If you really want to get into some new science, protozoan biology, if nothing else, protozoan phylogeny is really an exciting field because of the fact that we're trying to figure out these organisms we've lumped together in this group called protozoa simply because they're small. And we learned a long time ago that's not a good thing, just because way something looks. Let's look at the amoebas first. Let's look at the entamoebas. This is your typical amoeba, right? Lobe-shaped pseudopods. The little CV thing there, that's the contractile vacuole. So amoebozoans, many of them will have these lobe-shaped pseudopod, which they use for moving. This is how, when you're looking under a microscope, it's kind of hard sometimes for students to identify an amoeba because it looks like a blob, right? When they're alive, though, if you're looking through the microscope, you can tell it's a blob versus the amoeba because in amoebas, they'll have cytoplasmic streaming. There'll be movement occurring inside of them. Now, having a contractile vacuole tells us what type of environment this organism would live in. This is going to live in an environment in which it has to get rid of excess water, which, if you remember from Bio 1, is what we call a hypotonic environment, right? So a little bit of detective work there again. It's like with the paramecium. If you see a contractile vacuole in an organism, it's because it lives in a hypotonic environment. Gotta get rid of that excess water. Now, the pseudopods, like we talked about in lab, can be used for movement. This guy wants to move down in that direction. He just starts pumping cytoplasm. All right, hello. I'm sorry I got kicked off for a second, but I'm back on. Let me make sure the recorder's still on. Yeah, it's still on. All right, so if the amoeba wanted to move off to the right, it would just start forcing cytoplasm down into that pseudopod. If you remember from Bio 1, those actin filaments, those stretchy rubber band-like filaments that are part of the cell cytoskeleton, lots of those right underneath the membrane, making it very flexible so it doesn't burst when they're pushing out. Amoebas can also use this for capturing food too, right? They can use their pseudopods, kind of like the blob, and engulf food, paramecium, those kinds of things. Now, if everybody's really familiar with amoebozoans, you've seen them, we've talked about them, we've heard about them. But in this group, there are some really bizarre kind of critters out there. These things called slime molds, for example. Look at the color and the diversity. These are all living organisms, folks. Now, they're called slime molds because they make spores and they kind of look like a fungus. They are able to move. They're able to basically spread out across their environment. So they're kind of, movement is something we generally associate with animals. Spores, among other things, many different critters make spores, but so do fungus. So that's where we get the name up there, myctozoans. Myctozoan literally means fungus, and zoans is animal. I guess, actually that's mycetozoans. Got the accent in the wrong place. The mycetozoans, these are the slime molds. Again, not molds, only look like molds and called molds because of that thing we've talked about before, convergent evolution. Aren't those weird? These, I think, are really cool. Look like little golden eggs, don't they? Now, slime molds are really strange things, and there's two types of slime molds. First type is called a plasmodial slime mold. Now, before I go any further, let me make sure we don't get confused. There was a protozoan that we talked about earlier and you saw in the lab called Plasmodium, right? Plasmodium causes malaria. Plasmodium is the name, the Plasmodium falciparum, I think is the one we had, is the actual protozoan that causes the malaria disease. In this case, a plasmodium isn't referring to that critter. A plasmodium is it's a mass of cytoplasm, like up here in the upper left, that orange thing, that would be a plasmodium. So that's a different definition. Don't get those mixed up. A plasmodium is non-walled and multinucleate again. And again, many fungi do this. So another reason they were called molds, and I put molds in quotes to remind you again, these aren't really molds, right? But this whole idea of being multinucleate and having no cell walls, that's something, it's a very reoccurring theme that we see in molds. Synchronous mitosis, everybody divides at the same time. Boom, so that organism is getting bigger all at the same time. Now, plasmodial slime molds spend most of their life streaming along as a plasmodium. Okay, that's their feeding phase. They ingest bacteria and decaying organic matter, that sort of stuff. But whenever food or moisture is scarce, or whenever the environment changes, the organism then is going to start producing sporangia. And in sporangia is where we produce spores. In sporangia, these are sporangia, and these are sporangia. Now, if you think about it evolutionarily speaking, when food or moisture is scarce, why do you think that would stimulate this organism to start reproducing? Think about what we've said so far about evolution and how organisms deal with that. Why would a scarce environment, why would an environment that's getting tougher, or let's say an environment that is changing, why would that prompt these critters to undergo meiosis, to undergo this process of reproduction? Why would that environmental condition prompt that? Anybody got an idea for me? Why would these organisms begin to reproduce when the environment got rough? Remember, when the environment got rough, the environment is changing. How do species hope to perpetuate through a changing environment, especially when they don't know what it's going to change into? Remember, there's no way for organisms to be able to predict the future. Evolution has no goal. So what is it about these organisms that says, okay, when the environment is changing, turning into something I'm not sure what it's going to be like on the other end, I'm going to start reproducing? Why is that? What does that do? What does meiosis do? What's its purpose? It's got basically two purposes. What are they? Savannah, you had Bio 1 or Thalia? What's meiosis do? Daniela, can you help them? What does meiosis do? What's its purpose? Now, I know it makes sex cells. I know it makes sperm and egg, but tell me about its purpose as it comes to what we've discussed in evolution. You all can hear me, right? Okay, good. Okay, all right. Mitosis makes copies of cells, clones. Meiosis makes cells, but they're genetically different. They're variable. So when the environment is changing, when we don't know what the environment's going to be like, what organisms are going to do is produce all kinds of variability in the hopes that some of that variability will have an advantage, right? Go all the way back to what Darwin was saying. Why do we produce more offspring that can possibly hope to survive? Well, the whole idea is if you make lots of offspring, since it's all random how these genes come together, and when wherever they form chiasma, and how they line up on the metaphase plate, and how they separate, and what gametes, all that variability, when the environment is changing, you don't know what it's going to be. I mean, if you know what the environment is going to be like, you could evolve the correct variability, if you will. But if you don't know what it's going to be like, you're just going to make lots of variability, right? So in those times when the environment is becoming rough, food, moisture is scarce, the environment is changing, many organisms are going to start reproducing because they said, I don't know what's going to happen. I don't know if we're going to be able to survive. So I'm going to make lots of different kinds of offspring, hoping that at least a percentage of them survives. Okay? Now, sometimes organisms will begin to reproduce when the conditions are good, when they're right. Like, for example, many times with fungi, you'll find that the toadstools that pop up in your yard tend to pop up after a rainy event, right? Because those are great conditions for spores. So it's just kind of a balance, depending on how species respond to this kind of change that's occurring. Let's look at the life cycle of these plasmodial slime molds. Start at the top with the zygote. All right, so what happens is the zygote nuclei divide, but it stays one giant cell. It's multinucleate. So notice that this feeding plasmodium has several nuclei in it. Here's what it looks like. You go on YouTube and type weird stuff growing in the sewers of New York City. Just type that in. You'll find tons of videos of slime molds. People come across these things and they have no idea what they do. So our feeding plasmodium moves around as a multinucleate during harsh conditions, right? During harsh conditions, it's going to start and I am back again. All right, we're good there. So they produce the sporangium, and then here we go, right? Meiosis, whenever you see meiosis in a reproductive life cycle, variability. I think it's my pen. Anyway, whenever we see meiosis, folks, it's variability. Yes, meiosis makes lots of cells, but the important part is those cells are different, okay? And out of the sporangium, sporangia produce spores. They're going to make these very resistant spores that can last for a long time until they germinate. And if they find the conditions correct, then they will germinate, and they form these amoeboid cells. Now, amoeboid cells coming out of the spore, but we also have flagellated cells that come out of the spore, and those things come together and fertilization occurs, and they can switch between those two types. So flagellated cells and amoeboid cells aren't male and female, is what I'm trying to point out here, okay? Very interesting life cycle, right? But very much like ours too. Not so much like the alternation of generation thing, right? We've got a multicellular zygote, multinucleate one cell, okay? And then in the haploid stage is the one in the green, we're making amoeboids and flagellated cells that are haploid. So that's quite a bit like us. That's the way we do it. Now, there's another type of slime mold called cellular slime molds. These are really, really interesting, especially from a research perspective, because well, let me just explain what's going on. They have a very simple developmental system, and the reason why we study these is this is one of the ones that help us understand how maybe multicellularity blended into tissues. You see, tissues are differentiated. They do different things, right? They have a very specialized function. So what I mean by that, we start off with number one. In cellular slime molds, individual organisms move around as a separate amoeba. So that's an amoeba, just one amoeba. And they move through the soil, ingesting bacteria, using pseudopods like amoebas do, very much like we saw in the plasmodial slime molds. Now, here's something that's interesting. When food becomes scarce, the individuals begin to aggregate. The amoebas start coming together, right? And they grow bigger and bigger. And eventually what they do is they form this multicellular slug. Now, the multicellular slug then goes through a series of transformations where it actually ends up creating this fruiting body. Up at the top are where the spores are going to be made. So there's a stalk and there's a fruiting body. What's interesting about this is, as it comes to cell differentiation, these are all individual amoebas. When they come together and form that multicellular slug, as they're beginning to go through these steps here, where they form the stalk and the sporangia, different cells do different things. Different amoebas, cells, become part of the stalk. Some parts of the amoeba become part of the sporangial capsule. So there is cell differentiation occurring. So those amoebas live by themselves most of the time, but when they come together to reproduce, they come together and reproduce, and they begin to specialize. Like I said, some cells form the stalk. Other cells form the devices that form the spores. And then the spores, and asexual reproduction is very common in this species, right? The spores germinate into amoebas if it's favorable, which then move around through their feeding stage. And if they then aggregate back when the food is low and things are changing and things are getting rough, so I'm going to start producing some offspring, they go back and they start migrating together. Dictyostelium discoideum down below, that little thing that you see there, it looks like just a blob of nothing. That is a cellular slime mold. So these things engage in asexual reproduction. They also engage in sexual reproduction, right? In sexual reproduction, what happens is we get two of these solitary amoebas, and fertilization occurs and forms a zygote. That zygote then grows by consuming haploid amoebas. And then eventually that zygote, when it grows, is only short-lived. It only stays as a single cell. So this is a little bit like we were talking about the alternation of generation, because we've got quite a bit of the cycle is haploid on the left. You're only diploid for a short period of time. In some organisms, being diploid is just a means to an end. In other words, what I mean by that is being diploid is just a way to increase variability. Because you go through meiosis, and each cells you make, each gamete you make is going to be variable. And then the other amoeba would do the same thing. And then you mix those two together, and there's more variability. So you get, with meiosis and fertilization, you get like a double whammy of variability there. So you get fertilization gives you variability, then meiosis gives you variability, and then those amoebas move out into the environment and hopefully find a place to survive. There's a really cool video on Blackboard of the fellow who's done a lot of work with slime molds. Take a look at that. It's some really interesting stuff. The tubulinids, the next group. The tubulinids is a very diverse group. It involves your typical amoeba. Amoeba proteus is your very common amoeba. Amoebas are found in many places all over the world: soil, freshwater, marine. Some are predators, some are active predators, some are detritivores. Detritivores eat detritus. Detritus is decaying organic matter. If you were to dig down beneath the leaf litter in your yard, down into the grass, and you would get down to this spot where it's all kind of mushed together, it's kind of hard to tell what was what. It's kind of like an organic soup. That would be detritus. So a lot of amoebas feed on that too. And you can see in the picture too, they are also active predators, using their pseudopods to engulf things like like paramecium. Of course, amoebas can be problematic. Certain species of amoebas, especially what we call ent amoebas. Ent refers to inside. So these amoebas that are typically parasites. Many of these are parasites of vertebrates. Some parasitize some invertebrates. Humans, we host about a half dozen species of amoebas, some of them being pathogenic or disease-causing, like the ones you see on the page. The one on the lower left, Entamoeba histolytica, causes amoebic dysentery. Yeah. And then Naegleri fowleri up here. This is brain tissue, and these are the amoebas. This is the brain-eating amoeba that we've been talking about. So the brain-eating amoeba that moves up through your olfactory nerve into your brain, when it gets in your brain, starts to eat pieces of your brain away. But again, like I've said before, remember, most amoebas kind of do their stuff and leave us alone, right? So we've made our way down to the uniconts. The last thing I want to talk about is a type of uniconts called the opisthoconts. Now, cont means flagella again, or pseudopod. But in this particular case, we're going to be more specific about where the flagella is. You're thinking about, what do you mean where the flagella is? Remember, Euglena had their flagella on the front. Opisthoconts, opistho means rear, have their flagella at the back. My little sperm cell again. This flagella's at the back, right? He's moving in that direction. Now, the opisthoconts is even into itself is a polyphyletic group, right? It's got fungi, it's got animals, and it's got two types of protozoans, the nuclearids and the choanoflagellates, right? So quite a bit of mixing. That's why we call protozoans that stem basal group. All the other groups of the eukaryotes that we're going to discuss for the rest of the semester, the land plants, and the fungi and the animals, all of those obviously have arisen out of this sort of base or mix of protozoan organisms. All right, let's talk about protozoans in the big scheme of things on Earth, or what we call protozoan ecology. Protozoans act as many things in the environment. One of the things they can be symbionts. For example, and if you took my marine science class, this word probably sounds familiar to you. There are many protozoans, dinoflagellates mostly, that live in the tissues of, for example, coral. Coral reefs, the coral gets its color not from the animal. Coral is an animal, it's a jellyfish, related to jellyfish. But the color that we see in coral is not from the animal. It's actually from zooanthellae, which are symbiotic dinoflagellates usually, symbiotic protozoans that live in the tissue of the coral. You can see in the picture on the right, the zooanthellae are in red, and then the green cells are the cells of the actual animal itself. So these cells, these protozoan cells, these zooanthellae, live in the tissue of the coral animal. Now, the reason why that's a symbiotic relationship is the zooanthellae living inside the tissues of the coral. They receive protection. They receive a source of carbon, which they need to build up their own carbon molecules from, and the benefit for the coral, zooanthellae can photosynthesize. Right? So you get a photosynthetic meal. So zooanthellae photosynthesize, they make enough food for themselves, and the rest goes to the coral, which now you can understand why corals have to grow, or typically grow best, in clear, warm water, but in clear water and at shallow depths, right? They actually have to be at shallow depths in clear water to get enough sunlight so those zooanthellae are stimulated enough to undergo photosynthesis. Coral captures some of their food otherwise, but a big majority of the food comes from zooanthellae. And when we get into animals later on, there is a condition in which sometimes corals get rid of their zooanthellae. They actually expel their zooanthellae out of their tissues, and when that happens, many times they die. So this is what we call an obligate symbiotic relationship. Hypermastigote down in the lower right, that's a protozoan that's found in the gut of a termite. It's hard to believe, huh? Thinking about how big a termite is. But these things aid in wood digestion. Termites cannot digest cellulose any more than you and I can. Animals cannot digest cellulose, but there are bacteria and protozoans that can do that for you, symbionts, like those that live in the cow's stomach, right? Back in the 1860s, a protozoan called Phytophthora infestans caused something called the potato blight in Ireland. And this coincides with the mass immigration of Irish folks to the United States. When the potato blight hit in Ireland, it wiped out most of the potatoes. And in Ireland, there were quite a few poor farming folks, and without potatoes, they had little to nothing to eat. So with little or nothing to eat, a lot of those folks said, "The heck with," and they got out of the world, right? They got out of that part of the world and went to another part, with the United States. Protozoans are very important as producers. About a third of the world's aquatic productivity occurs by these protozoans versus on land, which is done by the green plants. So producers are those that photosynthesize, folks. Producers are the ones that produce food. So protestin producers as well as prokaryotic producers produce, they're the basis. They take photosynthesis, so they use it to make food, and along comes the herbivorous plankton or the carnivorous plankton, which then consume and feed upon these. And then, of course, you can work your way all the way up to the other consumers like the penguin and the killer whale. So protozoans are extremely important, especially in aquatic, out in the ocean, in forming the basis of the food web. Right? Because without producers, folks, in any ecosystem, no matter which one you're talking about, without producers, you have no way to get energy into the ecosystem. You and I, us animals, we can't photosynthesize, right? We don't have any way to do it. We have to eat stuff. Plants can photosynthesize. Protozoans who are producers and prokaryotes that are producers can photosynthesize. So they're going to form the basis of the food web. Now, keeping that in mind, how important it is to have that basis as a food web, this next chart's a bit disturbing. What we're plotting on the globe there is sea surface temperatures that have changed. I don't know, your book, can't remember what the time frame is. Let's see if it says in your book. Since 1950, all right, so in the last 50 or 60 years, this is the change in sea surface cellulose. Now, we don't need a lot of change in water temperature or air temperature to cause major environmental impacts. That's one of the misnomers about global climate change. Well, the temperature only went up a degree or so. With the sensitivity of the way ecosystems are on Earth, that doesn't sometimes take very much to cause problems. So, for example, in this case, as the sea surface temperatures have changed, in some cases they've risen. Like, for example, you can see down here, those have gone up quite a bit. Sea surface temperature is proportional to chlorophyll concentration. That's what that little word thing means. It's proportional. What I'm saying here is sea surface temperature and the amount of plants are related. The warmer the water is, the warmer sea surface temperatures, SSTs, the warmer the water is, the less photosynthesis you're going to have. The reason why is photosynthetic organisms need light, but they also need nutrients. And warm water, for reasons that have to do with the dynamics of the way water circulates in the ocean, warm water blocks nutrients, basically nutrient upwelling coming from the deep in the water. It blocks nutrient upwelling, so these organisms at the surface, they would have to be to photosynthesize, can't get access to the nutrients. So other than the southern Indian Ocean, all the other major oceans on Earth, the Arctic and the equatorial Atlantic and all the other oceanic regions have actually, you'll notice since 1950, diminished in chlorophyll. Diminishing in chlorophyll means diminishing in producers. Okay? Diminishment of producers. Now, those producers could be prokaryotes, protozoans, or plants, probably all of them together. But no matter what, that diminishment in chlorophyll means that there are fewer producers, and if there are fewer producers, there's going to be fewer food, there's going to be less food in that environment to support all the other members that come above the producers in the food web, right? So these guys start dropping in number, everybody else is going to be affected by it. That's why producers are some of the most important organisms in ecosystems. They're the ones that bring the energy into an ecosystem and allow it to be used by living organisms. The rest of us, as I said, we can't do that, right? We're at a slave, so be nice to your plants. Are any questions for me then? 'Cause that's all I have for that chapter. All right, I will stop the recording there. I will do chapter 29 when we get into class next week. Let me finish the recording.